So lots of research on pain has conventionally been done in animals, and we felt that some of the work that was currently being done on animals could potentially be replaced by a human model. So we developed this human neuronal model where we take dental pulp stem cells from usually wisdom teeth, and those wisdom teeth are going to be thrown away after extraction, but we take the tissue to the lab and then we do a tissue explant. Cells will grow out from that tissue, and then we can isolate stem cells. From those stem cells then we can differentiate these nerves that we call peripheral neuronal equivalents.
We're using this human neuronal model. It's going to be more specific to treating the pain and therefore have limited side effects. Positive of using this model versus animals is that if you had this undying desire to use animals, you physically couldn't, because this pain receptor is only expressed in humans. It's not expressed in animals, so you can't really use animals, and if you used animals then I wouldn't be confident the answer was 100% correct.
This receptor is found to be in humans to actually, when it is activated, it actually causes pain. But in animals, when it's activated, it inhibits pain. So you can see the difference now. We're talking about completely different action of the same receptor, because we know now, unfortunately, a lot of these medications fail to produce the desirable effect when they are taken forward to the clinical studies in humans. And that is simply because the neurochemistry of the animals is not the same as the neurochemistry of humans. That's why there is always the mismatch between the final outcomes when you test these drugs in animals and when you test them in humans.
We use that model then in the lab, and we can expose the peripheral neuronal equivalents to various stimuli, like various inflammatory stimuli, maybe stimuli like bacteria or viruses, and then we can see what happens to the nerves.
The current medications for pain relief are either not effective, that means they are not really very good in alleviating the symptoms of pain completely, or if they are effective in reducing the symptoms, they have very big side effects like the opioids, and we know the addiction associated with them. So there is definitely a need for novel targets for pain relief that can be as specific as possible to reduce the symptoms. Our project is investigating a receptor that is specifically expressed in the sensory neuron. That means we're reducing the off-target effect. The function of that receptor in humans differ from animals, so that hopefully then will bring us closer to a target that is going to be much more applicable to humans than to animals.